we're going to talk about what's new and what's hot in the literature. And I'm going to blow through 10 papers, leave time for questions. You can put the questions in the chat. There's only one cat paper. Sorry, it always ends up biased towards dogs. This year I tried to rectify that, and there were so many interesting dog papers and not a lot of database cat papers. But I'm going to talk about one of the database cat papers that is of interest to me because Katie and I have a project on this same subject with one of my graduate students and residents, Jen Vernick and Tracy Doucette in the psychology department here. So that'll be the first thing we talk about. And what I'm going to do is I'm going to sort of then ramp it up a little bit so that the hard stuff comes just before the middle and through the middle. And then there are going to be a series of papers about what we've learned about how we should be delivering veterinary care, because people have sort of finally caught on that maybe we should stop brutalizing animals and scaring them, and we might have some choices there. So I'm going to go through just a subset of papers that have appeared recently that have some interesting data. And I'm also going to tell you in these papers where there could be problems, where you could expect to see something different, et cetera, et cetera. The light is driving me nuts. So, okay. So this is out of Applied Animal Behavior Science recently. It's a 2023 paper. And it talks about the effects of early social separation on the behavior of kittens who have been, because social separation, separated generally from their mother. Now, that's part of the complexity of this paper. So let me just go through how they define what they did. They said they had 3 cat groups. The first group was mother reared, and they call this group the control. It had 32 kittens from a number of litters and an uneven sex ratio, which they will all have. The kittens here were raised by their mother together with at least 2 litter mates. That became the definition of mother reared. You had to have at least 2 litter mates. They didn't want a singleton kitten because orphan singletons is their last category. And they didn't want to have a 4th category of mother reared singletons, mother reared, you know. So what they ended up by doing is calling any 2+ kittens that were with their mother reared. Now, these were all cats that were found, and they were raised in a home rescue situation. So these cats had their own rooms. They saw people, people provided food, people provided care in the form of changing litter boxes. And in terms of the orphans, they fed them every couple of hours, but they weren't treated as pets, and they weren't overly cuddled and played with. So there were orphans raised with siblings, and then orphans who were singleton kittens. So there were 14 kittens from a number of litters, and these were kittens raised without their mother, but raised together as a group with at least 2 litter mates. Okay, so notice the 2 litter mates is a requirement for both mother reared and orphans raised with siblings. That meant that they got both interactions with a sibling group, not just one sibling, and their mom, if it was mother reared, or a sibling group, if they were orphaned. And they were hand reared from 4 weeks of age or earlier. And the mean was 3.5 weeks of age, which meant that they were fed. So they got a different type of human exposure than mother reared did. The orphan singletons were 16 kittens. And these don't really add up because they, there's a weirdness in this population. Not all of these were litter kittens. There were some kittens that were found. So there were 3 kittens that were found by themselves. So if you delved into how many were in the litters, it wouldn't add up, but that's why. So they had some that got lumped in a little later when these cats were found wandering around. And these are kittens raised without the presence of their mother or litter mates. Okay. So when they looked at the treatment groups, you had the mother reared, the orphans raised with at least 2 siblings, and the mother reared also had at least 2 siblings, and then you had orphan singletons. You found that the number of litters differed a little bit, the mean litter size differed a little bit, and this is where I told you 3 kittens were found alone, so litter size is, you know, they don't really know. The rescue pulled them off and made them into singletons, largely because of their body condition or what mom could do or the health of the mom. So there are a lot of things going on that they've lumped into early noise. The mean age at which the singletons were separated was earlier, and it's, you think 2.8, 3.5, what kind of a difference is that at that age? That's a big difference, actually. And they addressed that. So they had 3 behavioral tests: a struggle test, a meet test, and a separation or confinement test. Katie will recognize the struggle test because this is just what we call a holding test. The experimenter approaches the kitten, pets 3 times from the head to tail, picks them up, supporting their thorax and rump. And the test lasts for 30 seconds or until the kitten begins to struggle, and then the kitten's immediately put down safely on all 4 feet. The meat test is interesting. They take a piece of raw beef and they pre-tested these kittens to see if they would eat the beef, and they all did. They placed on a mat in the middle of the room, and it was on a string, so they could get it if they needed to. But once the kitten was chewing, the experimenter took bamboo tongs and tried to remove the meat from the kitten. The kitten had to have the carnassial pair gnawing on the meat. And they stopped trying to take it after the 4th attempt, or if the kitten didn't approach the meat for a minute. It's weird. The separation confinement test is interesting. First, the kittens underwent thermal imaging where their eyes largely were imaged. And then the cat was placed in a plastic carrier with a grill that you could see through. And then that carrier was placed in a room where they were completely left alone for 5 minutes. And then they were thermally imaged, and videotaping was involved in all of this. So the behaviors that they measured are defined here. So the latency to struggle, how long did it take them to put a paw out? Latency to chew, active defense or aggression. Did they swipe at the tongs or the human or stomp on the floor? Did they hiss or growl? When they had the confinement test, did they vocalize? Latency and number of vocalizations, and how long was any motor activity that they did? And then there were some interesting questions here, because when you look at the patterns here, this is the latency to struggle. The letters that are different are significantly different at the 0.05 level. Okay, and here they actually calculated the exact test. So it's 0.001. So the control moms who had at least 2 kittens had a much longer latency to struggle than did either set of the orphans. And there was no difference between them. This is not a statistically significant difference in this box and whiskers plot. After cleaning up the data a little bit for the non-participants, because when they got into the test situation, 3 or 4 cats did not eat the meat, they found out that the latency to chew for the kittens reared with mom was longer, and there was no difference between the orphaned groups, but there was also a statistically significant difference between each of these. And then they looked at the number of vocalizations that these kittens emitted, and the controls came up short, which shouldn't surprise you, because these kittens might be more secure if they're with mom. And the orphans raised with siblings and as singletons didn't differ from each other, but they differed from the control. And when they looked at the amount of time they spent moving in that cat carrier, the orphan kittens also moved more. So they were probably more distressed. And you can make arguments about a secure base, et cetera, et cetera. But it strongly suggests that they didn't deal new environments as well as the kittens who were with mom, because they vocalized more and they moved. So what did we learn? Control cats struggle less than did either of the orphan cat groups. Control cats had a longer latency to chew, but there were no differences in aggression. And that was an interesting finding, because I think all of us would have thought those orphan kittens who'd been on their own would be much more aggressive, and they weren't. The controlled kittens vocalized and moved less in isolation than either of the orphan kitten groups. There were no differences between singletons and the cats with kittens that were found in the orphan group, and thermography was not useful here. Okay. So that gives you an idea of how important mom is. And I mentioned that because we found the same thing. We didn't do these tests, but we found that mom was also important. Okay, so there was my token kitten paper. I was looking for something with hard data, and their data are hard. They're a bit messy, but they are hard. This is a very cool paper out of PLOS One, which is an open access journal, which means any one of you can download it. And this is called Dogs Can Discriminate Between Human Baseline and Psychological Stress Condition Odors, which is really important and interesting because people always say to me, well, do dogs smell fear? Do they smell stress? Well, I think they smell when you are worried. They probably smell some of the parameters associated with cortisol and sweat. And not everybody produces the same compounds, but that may not matter if there's something uniting the trigger of those compounds. So, and this is actually something that has been known in other circles for a long time and not able to be published. So what they did is they had the humans arrive. They set the humans up with some physiological measures. There was 3 minutes of silence, and at the end of the 3 minutes, they took a sample and they had a distractor for the dogs. And the participant also gave them some blank samples. The experimenter then administered an arithmetic test. So here's another case of math scaring half the population. And then after 3 minutes of the test, they took the physiological data, the same physiological data that they'd taken before, because these people are wearing all sorts of sensors. And then at the end of that, they also took the stress samples, and there was a self-report on a visual analog scale of how stressful the humans thought the mathematical test was. It was an arithmetic test. I don't know how hard this could be. That may have been in the supplemental materials, and I didn't pull it up. And then there was a debrief. So that's how they did this. And these are the samples, of course, that they are now going to give to the dogs. So this is the setup the dogs used. And this is a very, very, very standard setup for those of you who aren't familiar with odorant detection in dogs, where these are little canisters. They generally have a cap that allows odorants, volatile compounds to escape. They look very much like a spice tin. In fact, when we custom built an apparatus like this in Norway, we use spice tins, and we just fitted it to that because they were widely commercially available, so you could shake stuff out, and it was perfect for odorant dispersal. There are, and the nice thing is you can put the samples inside, they'll disperse, et cetera, et cetera. And then you can throw these in a commercial dishwasher and everybody's cool and you don't contaminate everything. There is a real design problem that was not addressed in this paper, and I'm not going to address it, but I want to just flag it for you guys. There are only 3 samples in this. There are end effects in these types of apparatus. So dogs will not necessarily treat each of these spaces as equal, and you need to test for that. And the other thing is, I can tell you right now that you would need a lot more tests than they did to really nail things down. But that's okay. They still had a large number of trials, and that's all that mattered. So here were the 5 dogs they had. Here are the number of testing situations, which would need to increase. Discrimination trials, except for Winnie, were pretty good. 40 is going to be a minimum. They don't distinguish here between the training and testing trials, and they really should do that. But the dogs ended up performing in the 70 to 100% range. The average performance across all tests was in the 90s. That's actually quite high. The mean number of correct trials was 18 to 19 plus, and they give you the confidence intervals. So these dogs at least could discriminate odor. There are some things that they didn't test for that they should have, given the way they set up that apparatus, but we will believe them. And the point of this whole thing is that while they didn't isolate the compound, 90 to 97% of the time, the dogs could tell the sample that was given before the arithmetic test from the sample that was given after the arithmetic test, and could identify when the humans were distressed. Now, that is extremely useful if you're going to have a service dog who either is going to help you with PTSD or possibly a diabetic crisis, or an epileptogenic event, because chances are you will start to feel a little distressed before those things. So this is actually a very, very useful thing. For those of you who are going to ask me, does this tell you, could the dogs use this to detect bad guys who are about to do awful things? Well, that all depends on how well they can control their emotions. And we have other tests for that, but they all depend on how well you can control your emotions. Okay, we're now moving into the harder terrain. This is a fascinating paper. It is in PLOS One. It's open access. Any of you could download and read this paper. This is called Network Analysis Reveals Abnormal Functional Brain Circuitry in Anxious Dogs. This is actually written by a friend of mine, and I need to disclose that. Kathalina Perimans is at Ghent, or Ghent, as she of course says, in Belgium. And I've worked with these people and helped set up some things. And in fact, one of the questionnaires used in this study is a version of our clinical questionnaire. So what they did here was they wanted to know how the brain circuits involved in anxiety differed in their function and their connectivity in healthy dogs and patients. And Kathalina has been working for years on imaging of the brain in behavioral concerns. She has measured dopamine transporter activity in aggressive dogs. She has measured serotonin activity and the effects of citalopram on both dopamine and serotonin. She's established the level of citalopram and escitalopram that you would need to take to be able to saturate receptors in various regions of the brain. And she's worked on using ketamine and MRI, the actual magnetic force of the MRI, in changing some aggressive dog behaviors. And there's a whole bunch of other things she's worked on. We've tried to work together on a number of studies. They tend to be very expensive to do. Now that the vet school has a T2MR, I'm looking a 2TMR. I'm looking forward to seeing if we can get Kathalina here for some studies, because we have the patient she's looking for. There is they struggle to get them at Ghent. There are behavior people there, but getting the people to participate in these studies, you've got to have somebody really committed to the studies. So they had 25 healthy dogs and 13 patients. They used a clinical questionnaire and CBARC to evaluate the dogs to make sure they were affected or not. And they used the CBARC scores only to look at the patterns and factors and circuitry. There were no global topological differences in the dog brains between the abnormal dogs and the normal dogs. And that's really important, because that means that, you didn't have a different volume in one lobe, you didn't have a different configuration of a lobe. So gross brain abnormalities were ruled out. Okay. And these dogs had physical exams, they had a neurological exam. They weren't showing any signs of brain disease or neurological problems. In terms of anxiety circuitry, local and global efficiency were higher, and the path length, the distance you needed to get to that, was shorter in the amygdala, which is the region required for learning fear. If you don't have a functioning amygdala, you can't even learn adaptive fear. But the amygdala is also affected in pathological fear and anxiety. And in patients, but not the healthy dogs, the local and global efficiency were higher and the path length was shorter for everything involving the amygdala. And if you looked at the connectivity between the regions, and I'm going to show you a picture that will nail this down in the next slide, there was higher connectivity in all parts of the anxiety circuit. In other words, the amygdala to the hippocampus. The hippocampus is the part of the brain involved in associational learning. So you've got the part of the brain that's required to monitor and exhibit and develop adaptive and maladaptive fear and anxiety. That's the amygdala. And the central nucleus of the amygdala is actually responsible for every single fear and anxiety sign any animal displays. And the cells involved in the central nucleus of the amygdala have actually been mapped and identified as to where they're located and which signs they affect. Okay, so this has been well studied. So you've got the association between the amygdala and the hippocampus. You react and you learn. The amygdala and the mesencephalic area, so you react and you integrate. The amygdala and the thalamus, you react, you integrate, you regulate. The frontal lobe, executive function, you put it all together. The hippocampus, where you learn. The frontal lobe and the thalamus, again, a regulatory response. And the hippocampus and the thalamus, so learning and a regulatory response. Different regions affected different suites of behavioral scores, and that's the important thing here. That is fascinating. So when we look at this, what you can see, if you look at the connections of the circuits, the anxiety circuits that differ between the patient and the control group, if you look at the yellow bars, that's where you get increased connection strength in dogs with anxiety. So between the frontal lobe and the hippocampus, the frontal lobe and the thalamus and the amygdala, the thalamus and the hippocampus. Thalamus is old brain. This is, by the way, this is all old brain. This is the neopaleocortex. This is, the rest of this is all old. This is the emotional circuitry that we all talk about when we talk about the limbic system. Okay, so the hippocampus and the thalamus, the hippocampus and the amygdala, the thalamus and the amygdala and the mesencephalon. The place where you have decreased connection strength in dogs with anxiety is the hippocampus and the mesencephalon. And that's pretty interesting because this has some integrative and regulatory properties, and you've got a decreased role for it to play here. And when they looked at the things that the regions of the brain and how they were affected, if you take a look at the key here, the control group is the black, the patient group is the blue. And if you look at characteristic path length for anything involving the amygdala, the frontal lobe, the hippocampus, the mesencephalus, the thalamus, the vermis, patients all had shorter path lengths. They were much more efficient because they had to go a shorter distance. Didn't matter which region of the brain you started with. If you look at global efficiency, most of these regions were more efficient, okay, in patients. And if you look at local efficiency, again, this is asking across all of these, is this significant? So for somebody who's going to say to me, boy, those frontal lobes and hippocampi look identical, that's because they are. But this statistical test is asking across all of these. You get greater local efficiency in communicating in the anxious patients. Now, what does that mean? And how do you, are we going to say that's adaptive for a maladaptive, a pathological condition? Well, what it means is that, and these are their conclusions. I'm just going to summarize the part that's important. Local efficiency is higher in the amygdala in anxiety-related dogs when compared with the healthy group. This indicates that there's more clustering around the amygdala, so there's better interconnectivity in the anxious dogs. And increased local efficiency also indicates increased functional segregation in the amygdala. Now that's important because what it says is that you've got that local integration and it's functional and it helps to segregate the different parts of the amygdala. That's the control over the central nucleus, where some cells cause you to shake, some cells cause your heart rate to go up, some cells cause you to pant or have an increased respiratory rate. Some cells would cause you to sweat, et cetera, et cetera. And then when they looked at the strength of the connections of the anxiety circuit, you have more efficient communication in the patient group between the amygdala and the mesencephalon and the amygdala and the thalamus, the regulatory areas. And they postulate that the increased functional connectivity of these three regions is in line with the well-accepted hypothesis of anxiety and anxiety disorders, that they're the result of an increased or an overactive functioning of the salience network, how you know and recognize something about the outside world. So they are hearkening back to the idea that pathology in fear and anxiety is a result of overactivity of the amygdala, that it reacts more quickly, that it communicates in a different way with different areas, it recruits areas very quickly, and that part of the pathology is your inability to turn that off. And this is an important paper because their claim is that they have data to support the idea that this doesn't get turned off. And what they then propose, and this is what I'm hoping to see if I can get Kathalina to work with us on, is if you use functional MRI, which she uses all the time, and I've also used, in patients where you absolutely have complete information with them. Here they were able to characterize abnormalities in resting state functional brain network topology that were associated with anxiety. Okay, so the technique will work for that. They found correlations between anxiety symptoms and network measures. Now, you have to realize they didn't assign these dogs diagnoses. In other words, the CBARC can't do that. It just measures factors. And they didn't have all the information to completely assign a diagnosis using the clinical questionnaire. So they just talked about anxiety, which we know can be many things. But here's what they would like to do. We'd like to investigate the potential of using functional MRI as a diagnostic tool for treatment response, such as pharmacological treatments or neuromodulation of treatments like the transcranial magnetic stimulation, where you would use that with or without something like ketamine. And here, what I think we will be able to do, finding the money for this could be a challenge, but we have enough dogs of, let's say, a generalized anxiety group or a hyperreactivity group of dogs who may not have had what they needed as puppies, or a separation anxiety group. We could certainly do the before and afters of those by diagnosis, by the scorable clinic questionnaire, and we could couple that with some genetic information. And I think it would be a very cool study to do. But I think this is very, very, very clever and very exciting, and it explains a lot. Okay. This is a paper out of Hannes Loewy's lab, and I know I've mentioned a lot of papers from his lab, and Mila Salonen, and again, friends of mine, and I served as her external examiner in our PhD committee, brilliant woman. They looked at aggressiveness, ADHD-like behavior, and environmental influence on repetitive behavior in dogs. Okay, and they did this using basically a version of our clinical questionnaires and the ADH scale. And here are the breeds of dogs that they looked at. Now, this is done by a questionnaire of 4,500 dogs in Finland. And they have managed to use that questionnaire was so extensive that they have managed to get half a dozen papers out of it, which is great. And that's all Mila's work. But they work their way from German Shepherd dogs to smooth collies. Lagotto Romagnolo are common European dogs. They have a lot of anxieties. They have a hereditary version of noise reactivity and noise phobia. And the probability of repetitive behavior was highest for German Shepherds. It was lowest for smooth collies. And if you look at rough collies, they're down here. Bull terriers are up here, and I mentioned that because they've been in a number of papers. Okay, so the herding dogs are actually distributed across this. Pembroke Welsh Corgis are here in their population. The Finnish population may not be like the European population, may not be like the Canadian or the US populations. But, and this is the thing that they do so well. So they ask the probability of repetitive behavior, regardless of what type of dog you have, dependent on whether or not this is a first-time dog or not a first-time dog. And first-time dog owners had more dogs with repetitive behavior. Now they defined that. And that includes dogs with OCD and just dogs who do a little bit of spinning. There's no differentiation here. Then they ask the probability of repetitive behavior if you're an only dog or you have multiple dogs. Only dogs exhibited repetitive behaviors more. And then what's the probability of repetitive behavior given how much exercise you get? Well, guess what? It's higher if you get less exercise. Now, what that strongly suggests is that many, many of these behaviors that people complain about are within their purview to help fix. Once you've segued into OCD, you're not going to fix it, but we use exercise, we use other dogs, and we use knowledge to change behavior. Why wouldn't that work if you didn't have a pathology? So this is just, to me, a wonderful example of how you use good history data to get patterns that are important to people. And it gives you somewhere to go with some of this stuff. Now I'm going to talk about the toughest paper that we're going to discuss. And this, again, is a paper by a friend of mine, Eleanor Carlson, who is an outstanding young molecular geneticist who is at University of Massachusetts and the Broad Institute. And this, some of you will have heard of this set of papers. They did an abstract for Science. They did a scientific abstract of their work, and I'm going to talk about that because it has some very cool stuff in it. And then they presented the big paper. And I am not going to do justice to that paper because I'm only going to talk about one subset of things. But this is a very cool way to do a study, and it's the way we're hoping to be able to continue the puppy study we're starting this summer. So Eleanor started something called Darwin's Ark, and Darwin's Ark is a project that collects information about pet, generally mixed breed dogs. That's what she was actually interested in. She wasn't as interested in purebred dogs. And she's a cat person, by the way. But where they can do crowdsource studies, you know, so if you can have the public out there help you, and that's exactly what happened here. They had tens of thousands of people complete these incredibly long questionnaires and send them a cheek swab. Okay, so they gave away their dog's DNA. There was informed consent and everything. And what they looked at here was biddability. And biddability is, you know, when I use the word biddable a lot, I have Australian Shepherds, okay? So I can tell you which one of my dogs is the most biddable. I can tell you it's Annie. And right at this second in time, the least biddable is that awful gray puppy you saw a picture of who's sitting at my feet right now. He's young, but he's also different from the other dogs. So I have a lot of variation in my household. When we think of a biddable dog, we're thinking of a dog who responds to your requests, who is willing to take direction, who's interested in doing this, and then executes this quite well. And they had almost, I think, they had over 10,000 dogs for this. And they ranked dogs on the basis of biddable, and I'll show you a blow up of this, biddable and independent. And the independent dogs, and I know this is going to be a shock, were Basset Hounds, Alaskan Malamute, Shiba Inus, Miniature Pinschers, Great Pyrenees, Siberian Huskies, Beagles, Westies, it sort of surprised me a little, Dachshunds, and Yorkies. Now, the biddable dogs were Belgian Malinois, Vizslas, Border Collies, Australian Cattle Dogs, German Shepherds, Aussies, Goldens, Berners, Catahoula Leopard Dogs. I love Catahoula Leopard Dogs. And Labrador Retrievers. Now, let me explain their color coding and sign system to you because it's going to come back in a minute. So for every factor they looked at, there would be two colors. So biddable would be one end of the spectrum and independent would be the other. Within those color codings, the strength of the effect is the size of the circle and the intensity of the color. Okay. If they have a positive inside the circle, it's significant at the P equals 0.05 level. If there's a negative in it, it's not significant. And if there's a 0 in it, there may not have been enough dogs to test. There's nothing. If they have absent data, it tends to show up as a gray, and I don't think there are any circles that are gray here. And then they did a permutation or a probability score, and here you can see that -4 goes towards this end, which in this case is biddable, but it would be any factor. And +4 goes in the other end where they're independent, and then 0 would be they're neither. So they've taken each of these factors and stretched them out so that in the middle, you wouldn't be on one side or the other. And then the strength of the difference of the effect is relative to the size and the color density of the circle. Okay, if anybody has any questions, let me know. So when they looked at all dogs and they looked at where they fell, 95% of them fell with biddability. Here's the median score, okay, and here's the distribution. They looked only at border collies. The median score was further towards the biddable, and so was the distribution. Now, when they looked at border collies that were border collies, and they confirmed this by DNA, you find a bunch of dogs over here at -2. And here's this one. God knows who's over on the independent side. But if you look at mixed breed dogs with border collie ancestry, again confirmed by DNA, what you find is that you've skewed this to the side of biddability. And you can look at the density of this heat map so that you've shoved them onto the biddable side. That's what this density of color at the bottom is. And when you do this, this is a genetic effect of having border collie in your ancestry. So it makes you a little more biddable most of the time, not all of the time. Okay. Now, in the main paper, they actually asked an important question, and they asked how much of what we see in behavior is heritable and how much is due to the breed. Now, I just told you that this is the distribution of border collies, and this is the distribution of dogs with border collie in their ancestry, and it skews to this side, but it's not deterministic. Okay. What about for other things? Well, they looked at heritability, and heritability, or H squared, is a measure of the amount of additive genetic variance, the variance on which we can select. This is the variance that will allow you to make cows that produce heavier amounts of milk. So, they may produce an extra kilo of milk a day. This is how you select on butter fat content in milk. It's how you select on marbling in beef. How much variation is there in how much milk is produced, how much butter fat is in it, how much marbling is in the cattle, in the population in front of you? And their population were their samples. So they looked at heritability, the amount of additive genetic variance over the entire amount of how different the population was, what it looked like. The whole population of cows and buttermilk and poundage or kilos of milk they produce, marbling, height in dogs, and if you look at wolfhounds, they're a little different than Chihuahuas. So the dog population as a whole, or just the wolfhound population. So they were looking at all of their dogs. And when you see additive genetic variance over phenotypic genetic variance. That tells you how much, that's H squared. It ranges from 0 to 1, so it's a fraction. And it tells you how much selection you could do to change that factor. So an H squared of 0.5 means that half of the difference in all of those phenotypes is genetic, and you can select for that. You can selectively breed for that. It tells you nothing about the number of genes involved. It tells you nothing about the strength of the genetic effects. It's simply a quantitative measure of how your population varies. You bring in a whole bunch of new sires next year, you're going to have a different H squared because they're not the same population. And it can change with selection or inbreeding as populations change. And the inbreeding factor in purebred populations is important because there is a popular sire effect. So if you were to look at black labs, for example, in North America, relatively few sires contribute to most of the breedings, given that it's among the most populous breeds there are. And that means that you don't have the same variation you would think that you would have. So heritability changes very slowly. If multiple genes are involved, if one gene's involved, it can change quickly. But it gives you an idea of how much effect you could have by breeding, by selecting for that trait. And it's being used to decrease hip dysplasia. So if you look at how lax hips are and you measure dogs using a standardized measure, it doesn't matter if it's the British one, the Cornell one, the Penn one, you stick to one good quantitative measure. In 5-10 generations, you can completely change how tight the hips are in Rottweilers or Golden Retrievers or German Shepherds. And we know this because it's being done. Okay. If you don't allow breeding below a certain score, then you will change the population. So when they looked at these dogs, they found something that surprised them, but did not surprise me. And in fact, before they did this, Eleanor said, "What do you think is going to happen?" And I told her, and it's exactly what happened. So physical traits have a very high heritability. Hip scores, how tall you are. Physical trait related things, stride length, okay. Motor patterns, whether you run or you lope, Bassets versus coursing dogs. High heritability. The factors that they looked at. So the various factors they looked at in their behaviors. Whoa, the main heritability came down here at about just a little over 20%. And then if they looked at other behaviors, just random behaviors that were in the questionnaire, they came in at about 24%. So the bottom line here is that, and that you can see what the statistical differences were between the physical trait and behavior, physical trait related and behavior, motor pattern and behavior, and the factor in behavior. And the factor in behavior was not significantly different. And that shouldn't surprise you because behavior is behavior. Okay, now what that tells you is that there is some space for selection for behavior, but there's a lot less than there is for anything physical. And when you look at the patterns that they found, here were their factors: human sociability, arousal level, toy-directed motor patterns, biddability, agonistic threshold, dog sociability, environmental engagement, proximity seeking. Are you affectionate? Are you aloof? The only thing that they really found a pattern for is if you come down here for 3, they got some highly significant results for whether you would play with toys. There were missing data. There are the gray boxes. There were non-significant things, but they didn't get the power that you saw for biddability across the board. Here's biddability across the board as a factor in the way we dissected it before compared to the mixed breeds. So when you look at this, they tell you Human sociability is quite heritable. The toys and biddability are heritable. Everything else is wifty in this population. So here's their discussion, and let me summarize it. For 110 questions, 8 factors about breed explains more variance than does size, age, or sex, but the effect is very, very, very small. Breed correlates or is associated with heritability and has 5 times the effect for physical characteristics than it does for behavioral ones. So all this stuff about, oh, of course he does that. He's a German Shepherd. Of course he does that. She's a Border Collie. That's nonsense. Age explains little variation except for 2 factors, arousal level and toy-directed motor patterns. Well, that makes sense. You know, young dogs. Sex is associated only with lifting a leg to urinate. And the conclusion they come to is we make a lot of assumptions about breed that are not substantiated by data. Dogs really are themselves, and very little is deterministic, although the historical usage of breed will always shape behavior. And that's what I told Eleanor. And it shapes behavioral pathologies. But I told her she was likely to get as much variation within a household of dogs of the same breed that came from different sources as she was to get in her overall population. And it looks like that was borne out. So we walk around with a lot of myths. Okay, that was hard. I know all that was hard, and I only scraped the surface, but I want people to share some of my passion for behavioral genetics. I want people to understand that that type of knowledge is power, and knowing just a little bit and knowing it well can help you to help people, because misconceptions, I think we can see in this day and age how misconceptions, especially about science, get us into real trouble, and we can just do better. So I'm going to wrap up by reviewing a few papers on cats and dogs who receive veterinary care. It's a paper that was just published recently in JAVMA, and it's a placebo-controlled double-blinded study on using a single dose of gabapentin for dogs who are going in for veterinary visits. It's again by a friend of mine, George Moore and Nwako Ogata are both friends of mine. 22 healthy client-owned dogs, 1.5 to 8 years. They gave them 50 milligrams per kilo. Now, I want to applaud them for that, because we go up to that dose, and yet our medicine service, when they see that we've given a dog 35 milligrams per kilo every 12 hours, gasps. 50 milligrams per kilo of gabapentin or placebo 2 hours before a 5-minute standardized examination protocol. They measured behaviors from video. They measured salivary cortisol. And now you're going to see eye surface temperature, infrared measures come back, thermography come back. So here's how they decided on who to use. They assessed 34 dogs for eligibility. They had to exclude 12. This is exactly what happens in these studies. By the time you're done, you get nothing left. They randomized the remaining 22 that they had. 11 went into the gabapentin treatment group and 11 went into the placebo group. And the only reason that's even mentioned is that that's small. Okay. So they had a pre-physical examination phase in the waiting room, physical examination phase in the exam room, post-physical examination phase in the waiting room. And they timed these, which was good. They did a greeting test, an eye surface test, and salivation at 9 minutes just before they went into the examination room. And then they did it just after they'd sat in the waiting room at the end again. Okay. And the only significant finding was for lip licking, truly. Dogs sat less if they were given gabapentin, but lip licking is the only thing that popped up. And when you ask, what did we learn? Well, there were no adverse events associated with 50 milligrams per kilo, which is great, using gabapentin. Only one significant finding, decreased lip licking in the gabapentin group, suggesting that it affected an anxiety-based sign, but not a whole list of behaviors. And considering the measures and their sensitivity, infrared cameras in dogs' faces, lower sensitivity for salivary cortisol, et cetera, I might redesign this study. You're sticking an infrared camera in a dog's face. I think that's probably going to freak them out anyway. Salivary cortisol only produces 4 to 10% of the concentration as does serum cortisol. Now, you have to stick a dog for serum cortisol. I get that. But you're up against that limitation. So you've really got to have dogs with a lot of spit, and you've got to have a profound effect, and you may not get it in a small sample size. So here's a case of brilliantly designed study, did not produce what they hoped. I might change the study design, and I might get a heck of a lot more dogs. This is called At the Heart of a Dog's Veterinary Experience. Heart rate responses in dogs vary across a physical exam. And this is the first of 2 papers from this group I'm going to talk about. So they're fitting this dog with a cardiac monitor. Okay, so this is a monitor that can measure heart rate and heart rate variability. Here's the room where they're going to use in the exam table. This is all staged, nicer rooms than we have. And they start by petting the dog. They take the pulse. They listen to chest sounds. They palpate the belly. They lift the right front leg, the right back leg, the left front leg, the left back leg. They look in the right ear, the left ear. They fake taking a temperature. They look in the right eye, the left eye, the teeth, and then they do a mock vaccination. And every single place where you see letters, you have the association of differences. So A here is different from C, B, C, and D everywhere you see that. Okay, so they got a number of statistically significant differences for the parts of the exam. And when you look at the trace of the heart rate for the parts of the exam, you can flag the various parts of the exam in the heart rate. Dog 35 was a border collie who was actually pretty calm. That's the dog down here. Dog 17 was a greyhound who actually became tachycardic in a couple of places. And B here is picking up a right front leg. We have a paper that is soon, I think, going to be accepted, where the resident showed that picking up a leg can be the end of an exam. So I'm not surprised by that. The dog was already aroused, came back down a little bit, but nowhere near baseline, stayed aroused, and then you stuck something in his ear. Okay, and he reacted to that, or you pulled on his ear. So what did we learn from this study? Even mock exams are upsetting to dogs. And, you know, stop there. It's a major finding, and until somebody understands what it means, they should not be allowed to touch a dog. What does this tell you? Even if the intent isn't there, even if you're friendly with the dog, even if you're playing with the dog, putting a dog through a procedure is upsetting. It's going to change every one of your parameters. Why don't we teach dogs to do the procedures? Patting, the first step, and mock vaccination, the last step, were the most distressing parts of the exam for the dog. Approximately a third of the dogs had heart rates above 180 beats per minute at some point. The greyhound was an exception at 203, which means their heart is not adequately filling. And the proportion of time the dog spent with the tail tucked between the legs was predictive of the heart rate. Not greatly predictive, but 45%. The time is long past that we should be teaching dogs to not just cooperate, collaborate. Show me how you want me to do this. Here, if I ask you to turn your neck, can you turn your neck? Can you lift your elbow so I can get a stethoscope under there? Can you lie down on your left side so I can listen to your heart on both sides? How about can you stretch out and take a deep breath so they can take a chest radiograph when we say breathe? Can people do this? All of my dogs do this. We teach them. And this is the difference. So the same group looked at puppy pedicures. You're going to recognize the setup because it's exactly the same. Heart rate monitor, and now they're clipping dogs' toenails. These are the behaviors they looked at. They looked at tail posture, ear posture, what the mouth was doing, whether it was closed and relaxed, whether it was closed and tense, or whether the dog was panting. And when they looked at their model that explained the variation of heart rate observed when they did these clips, and they had fake clips and real clips, and there was a whole clip procedure that we didn't go into, a couple of things really mattered. Being female mattered, and the sum of the stress behaviors mattered. So when they added up all their stress behaviors, that affected heart rate. So that was a measure of what the dogs were telling you. And you can see that heart rate by itself is not a predictor of whether or not they'll accept the nail trim. If they had a previous painful experience, that is a good predictor of whether they'll accept the nail trim. They won't. And if they have failed in at least one step of the nail trim procedure that they set up, that's a good predictor that they won't cooperate with it. So these are important things for us to learn. So what have we learned? Heart rate during nail trim was higher than during baseline. Mean heart rate for female dogs was higher than for male dogs during nail trim. Refusing to take a treat from the examiner was correlated with having a painful experience in the past, and painful experiences in the past and failing one of the training steps was enough to say that you would have a problematic nail trim. What about the effect of owners when their dogs are there, presence or absence? And this is Florence Gonnet's lab in Paris. It's actually Toulouse, but she's from Paris. And here you see the dogs on the table, and you can decide about the table and the dog. But this is a common way dogs are examined. This is how dogs are examined at AVC, except the client may be there for some part of the exam or may be there for none of it. And certainly once laboratory evaluations are involved, the dog is taken away from the client. So they looked at the amount of restraint necessary for these dogs, and they looked at low, medium, and high. And this is a dog with low restraint, a hand on the chest, a hand on the rump so he doesn't slip off the table. And this dog, I hope you appreciate, is very scared, broad stance in the back, staring at the human, ears back, tail tucked. And they looked at whether they could put them on a table, they could look at their eye, their ear, their mouth, palpate them, skin folds, sculpt them, thermometer, move their paws, a success or a failure. And on a scale of 1 to 5, they evaluated how tough it was. And they looked at all of these things when the owners were present and when the owners were absent. So when they looked at entering the room, how stressed was the dog entering the room if the owner was present? And this is a criticism of this paper. They used the same type of scale, but they have them going in different directions. So let me explain this. For entering the room, a high score means you had to drag the dog in. A low score means that a low score means you had to drag the dog in, and a high score means they entered voluntarily. That's exactly the opposite of our scores. For the emotional state, a low score was relaxed and a high score of 3 was not. So that direction, although scoring system differs, that direction is the direction pretty much everybody's using. This one is not. So a high score here is a more relaxed dog. So just entering the room, the dog had fewer behaviors indicative of being distressed when the owner was present than when the owner was absent. And their emotional score was lower when the owner was present than when the owner was absent. So what did we learn here? When we think about the limitations of this study, there were no male owners, no dogs that were huge, no old dogs or puppies, no aggressive dogs, 25 dogs per dyad, minimally invasive exams, restricted owner-dog interactions, in other words, highly controlled. This study showed us that the presence of the owner in the veterinary consultation helps to reduce the stress of the dogs. During the examination phase, the dogs looked at their owners and appeared to seek social information from their owners. And if their owners were gone, they looked for them. Seeking social information is critical. It's how dogs learn whether there's a threat. And behaviors such as greeting the owner and door-related behaviors suggested, even when there were no other differences, the dogs were looking for their people. They felt the stress of being left, and it mattered. And I'll come back to these questions at the end. I see Amy's posting some questions, which is good. So these, even if they weren't showing acute behavioral differences, they were showing signs of being distressed. that were just associated with looking for their people. And the final paper I'm going to talk about is a paper that both Katie and I are an author on. And I'm going to talk about it simply because it takes such a different approach, and I just want to let you know about it. Again, it's in an open access journal, and it's called, it's in Animals, Effects of Changing Veterinary Handling Techniques on Canine Behavior and Physiology, Part 1, The Physiological Measurements, and it's by my resident and graduate student, Camille Square. And we had 28 dogs, 14 in each group, in a control treatment and an intervention treatment. And we looked at differences in how we treated the groups. So for the scale, in the control group, you were weighed before the exam and you walked on to the scale. In the intervention group, you were weighed after the exam, and the scale was covered with a blue non-slip yoga mat. It was moved away from the wall, and food treats were used. And this was done in a staged way. The physical exam, in the control group, you wore a white coat. Small dogs were always on the table. In the intervention group, no white coat. All dogs were on the floor or the client's lap, unless they said the dog prefers the table, in which case they got a mat on the table and the client. And we used licky mats and a non-slip blue mat that they could lie on for the examination. And licky mats had cream cheese on them. For the blood draw, we applied fake lidocaine to 3 of the legs. And why did we do 3 of the legs? It was the maximum number of legs we were willing to try for blood. 2 sticks, 3 legs, there you are. Standard needle and syringe and standard restraint. For the intervention group, we used real lidocaine. We used a closed end butterfly catheter system that meant that we didn't have to hold off the vein. We just had to get it into it, and it's closed system. It'll just draw the blood on its own, which is what you're used to when you have blood drawn, and reduced to no restraint using guidance and positioning. And then the clients had homework. The control group petted their dog for 5 minutes 3 times a week and kept notes. And the intervention group were given the set of the exam steps that we wanted the dog to learn to practice as part of just massaging the dog. And they did that 3 times a week for 5 minutes. And what we found is that when we looked at the ANOVA for overall changes across 4 visits, baseline, visit 1, visit 2, visit 3, we found that there was no overall change across the visits, okay? Now, this is the control group, this is the intervention group. But when we compared the first and the last visit, there was a massive change in serum cortisol for the intervention group. So what we found is that for both groups, there was an increase in serum cortisol in the second exam visit here, and it was followed by a decrease at the 3rd visit. And that may explain an initial period of sensitization followed by habituation. We can't rule that out because we saw the dogs every 2 weeks. And the pattern of serum cortisol over time, where it seems to start coming down, suggests that there could be habituation. So if you practiced over time and didn't do much else, you could probably teach these dogs to be calm. But we did something else. We created a standardized summary variable for stress, and we called it the stress response index score, where we combined all of our physiological measures because we had a bunch that weren't significant but were going in the right direction. We combined everything. We standardized them so that they were all on the same scale, and we Winsorized the data, meaning we removed the outliers. And then we did the statistics on that. And this is a standard procedure. And we found something very interesting. Here's the intervention group at the first visit, the baseline visit. Here's the intervention group at the last visit. Here's the control group at the first visit. Here's the control visit group at the last visit. It appears that the control group is actually getting worse. If you combine all the measures, including heart rate, neutrophil lymphocyte ratio, et cetera, with cortisol. So we're improving, big statistical difference, and they're not, which means we may be worsening dogs at every visit. So the effect size of this index was 1, suggesting that the effect is real and possibly important. The non-significant increase in the stress response index for the control group suggests that we probably do sensitize dogs. And we don't use these composite scores often enough, but we will now. So what did we learn? Veterinary examination designed to minimize stress and fear that was collaborative and adaptive to the dog's needs had a greater reduction in distress across 4 veterinary visits as measured by cortisol and the composite stress scale. The targeted low stress interventions were simple and practical and were specifically selected so that anybody in any practice could use them. And no one parameter was emphasized. The entire non-compartmentalized experience for the dog was what we were assessing. We were assessing what the dog experiences, not the pieces we do to the dog. The dog experiences us and everything else. And the physiological measures support that the dog viewed the 2 styles of examination differently. We can't attribute that to any of our changes, but the exams were viewed differently by the dogs, and that matters. Okay, so let me answer the questions. And in the second study, how could the researchers tell that the dogs could ascertain that certain odors stressed humans versus just being different than other pre-test odors? Okay, so what they did was they had that human's odor. That was the first swab test. So they had that human's odor at rest, and then that human's odor after the mathematical test. And they had a bunch of distractors of room and chair and other odors that the dogs didn't react to. And they repeated that 3-test design multiple times in a randomized way. And the differences then were only between the first odor that was given when they were calm and the last odor when they were distressed. With respect to petting at the beginning of an exam, can increased heart rate ever be associated with positive psychological state, anticipation of treats, play, etc.? I imagine it could be, but when you realize that they were also monitoring the facial expressions and the body expressions, that's what you look at. So it would be interesting to see if the cardiac changes with increased heart rate associated with play behaviors. You could do this. You could put these monitors on dogs and then go play with them and find out if the pattern was the same. Nobody's done that. So, Lisbeth, I'm going to see you soon. So during the pandemic, the dogs hated being without their owners and the vets loved being without the owners and thought the dogs loved it too. Oh, great question. Brian, by the way, said the same thing. He said to me, I don't know how you're holding up in the pandemic, but I'm loving it. Dogs are so easy to handle. 2 questions. Are they afraid? I don't know whether we assessed for that. They're missing their people. And, people would say, well, they're not acting up because the people aren't there. Well, there's another factor that nobody evaluated. There's always a performative part of examining a dog. The client is there. You're trying to do the medical stuff. You're also very aware of how you're coming across, or I think maybe I'm very aware. I'm very aware of how my residents are coming across. I'm always making sure we're communicating. And, when I go to interact with the dog, I'm in a different zone. So we're only in one zone. We're doing it at our pace, not the client's pace. So until you can tell me about all of those data, I don't know. I always want the dogs to be with the people. I think they're calmer. And the data are beginning to show that. Certainly my dogs, you may get something done, but they're harder to get back in if you take them away from me. They don't want to do that because it scares them. I have a few questions. From the paper about anxiety of dogs, I know it's a newer study, but are there any linked studies about applying neuromodulation to the overactivity noted in anxious dogs, or is this in the works? That's in the works. Kathalina and her group are very interested in that. In one of the later studies, there was mention of mock exams being upsetting to dogs. Have any studies been completed on happy visits, victory visits, and the impact of these on stress levels? Well, that's what Camille's study was about. I mean, basically, those were physical exams and we took blood. And that at least gave you a range of things. Just happy dog, I'm coming to the vet to learn about these things. No, and somebody should do that. That would be a very useful study. And if I were to design that study, I would recommend that you used a series of happy dog visits for each dog. And you had a standardized way you were going to look at this. And then you applied those same techniques to a series of real visits. because these one data point things are annoying beyond belief. Slightly unrelated, on collaborative care and training a dog to accept a stethoscope placement, other parts of the exam. Do you have links or resources? We need to develop them. We need to develop them. And Camille's out flat right now. I'm sort of out flat. We really do need to develop it because collaborative and adaptive care is very different than cooperative care. I have some videos. I don't know whether Camille has posted them on any of our websites. We have a YouTube channel. I don't know. I will find out. What I was trying to ask you, my first question is, did the dogs know that the person was stressed, or just that they smelled differently? Oh, they didn't meet the dogs did not meet the people. They met their scent. It's the only way you can do these tests. Sorry, I misunderstood Amy. Anybody else? Nobody, everybody's fine. Email if you want to reach out to me. Somebody has reached out to me for history forms. I'm hoping to get to those this week. I'm back in the States for a little bit, and it's just been a little crazy. The journal website, if you want appointments, you know about the seminars, they're done. And I think more and more people are asking about behavior at the beach. So I think it's going to happen again next year. And I'm done.